Um, Hi, thank you very much for the organizers for inviting me. I could watch some of the talks today. The first talk was at 3 a.m. my time in New York, so I'm afraid I missed some of those, but what I've seen so far is really interesting and exciting. And in my lab, I have a lot of projects that I could have talked about. I had a hard time deciding. But I finally decided to share with you our latest project, and it has to do with the mechanisms behind visual curiosity in particular. It's a paper that we just submitted for publication, and this is my first presentation on it. So let's see how it goes. So I can share my screen. Okay, can you see my screen? Okay, I'll assume that's a yes. I didn't hear, but I'll assume that's okay. Okay, so this project, thanks, Matthias, looks at investigates the neural pathways that give rise to curiosity, and in particular, perceptual curiosity and visual curiosity. We're investigating using fMRI in humans. And before I begin, I want to give I want to introduce, yes, the first author of the project, Michael Humpor, a fantastic graduate student in my laboratory. He was co-supervised by me and Mariam Ali in the psychology department. And I want to give credit to general support at the Zuckerman Institute and to NRSA funding for Mike. So I think that we all know by way of background that intelligent animals like humans have faced this conundrum. We act in very large, very complex environments that highly exceed our computational capacity. And we have to learn and discover regularities in this environment. And learning is a very costly process, takes a lot of energy and resources. And also, we can never be perfect at it because we face this conundrum. We have to select what to learn, decide what to learn, but we have to do that while we are in a state of ignorance. So we have to select what we learn before we know if that learning will produce anything useful or if it's worth it at all. And so there is general consensus in the field that this kind of conundrum requires a system of intrinsic motivation, which very broadly speaking says that there must be some internal state or states in the brain that motivate us to take actions and expand energy before we know what those actions will produce. And this intrinsic motivation most likely comes from previous knowledge. So we are not tabula rasa, we come with knowledge, a lot of previous knowledge, we know a lot of things. Some of those things that we know have a value. So this is previously learned, so we have reasons to anticipate that some things will be valuable, and that motivates us to take actions and seek rewards. Other states, however, have uncertainty. And studies of information seeking and curiosity tell us that uncertainty also motivates us to take actions independently of value. And this is what we call curiosity or information seeking actions. So in neuroscience and psychology, there have been scores and thousands and tens of thousands of studies about the mechanisms of value, how value-related constructs motivate actions. But we know much less about this other state of uncertainty. How is uncertainty so you have a state that has this property called uncertainty. How is that property represented in the brain? And how does it become motivating? How does it motivate us to do something, to expend energy and take certain actions? So this is the big topic that we wanted to explore in this study. And we started by thinking about visual uncertainty, because the visual system is the system in which we have the most developed ideas about how uncertainty might be represented. And in the visual system, we think that uncertainty is sort of a broad, representations of uncertainty are broadly distributed, and they consist of at least two processes that have been called first order and second order uncertainty. So first order uncertainty is a form of uncertainty that's embedded in the lowest level sensory representation. So here, even in visual cortex, primary visual cortex. As you know, primary visual cortex represents elementary visual features. And as I will show you in a moment, that representation can convey more or less uncertainty about those features. And that representation is called multivariate because it is conveyed by a population of neurons in visual cortex. I will explain this in more detail in a moment. And then, but we also know that many other brain areas at higher order cortex and also subcortical structures encode what we call confidence. And confidence is a self-reported rating. How confident do I feel about something? And that's also a statement about how uncertain I am. So confidence, the more confident I am, the less certain I am. Confidence depends on sensory factors in part, but also depends on context, response biases, all kinds of other behavioral factors. And these representations of confidence are univariate in that they're a bit simpler in that it's simply if those areas respond more, you are more confident. So it's a one-to-one relationship with the average response in an area. Okay, so let's look a little bit at this first order uncertainty, multivariate uncertainty. So I think many of you are familiar with this. This is, we can look at primary visual cortex. So we know that primary visual cortex represents the world in a way that's very detailed and not very intuitive, not exactly corresponding to what we phenomenally see. But so there is a retinotopic mapping in primary visual cortex of different locations, but also neurons encode elementary visual features. So for every location, you have specializations of neurons, like I'm showing here in this cube, where different local populations of neurons are tuned to different elementary features. One of the best studies is orientation. So for example, here, if you look at any location in the visual field, you'll find neurons that respond best to a line that is horizontal, slightly tilted, with very fine gradations of orientation. Okay, so now imagine that you see that I show you two pictures in the external environment. I show you this grating, which is horizontal, and I show it to you in a way that is very clear to you. You have very little uncertainty here on the top, or in a way that's corrupted by noise here in the bottom gray patch, where you really are not so sure. You have to think a little bit about what orientation it is that you're seeing. So there's a lot of physiological evidence that these percepts, that when you have a very clear picture with low uncertainty, this evokes a strong activation in a small population of these primary visual cells. So cells that are precisely tuned for horizontal lines will respond very strongly, and cells that are tuned to other orientations around them will not respond. In contrast, if you show a more fuzzy image, the pattern of activation will be broader. So the peak activation will still be at the true orientation, but the cells at neighboring orientations will be more uncertain, so to speak, about what it is they see. And they'll say, well, maybe the grating is a little bit tilted, and they will respond a little bit more. So you have a more diffused. So in other words, this sensory representation if you have low uncertainty or a clear visual percept, it will be encoded as a strong focused activation. Whereas if you have a diffuse visual percept, an unclear one, it will be encoded as more diffuse, broader activation across the population of neurons in this primary visual cortex. And so there has been a lot of beautiful physiological and computational work showing how these differences in the population activity really convey the uncertainty about a visual percept. On the other hand, there have also been very beautiful studies showing that when people, and these are in tests in which people are asked to provide ratings of how confident they are about what an image is, there are many areas that scale. Hello? Okay. There are many areas whose average response scales linearly with the confidence ratings, right? And this is in a variety of tasks that use verbal material, visual material, and so on. And again, these are many areas, and the areas that show this kind of response can differ by task and by context. Okay, so the hypothesis then in the field is that we start from a first order certainty or uncertainty that's encoded already in the bottom-up sensory input. That population representation is read out by these higher order areas that encode confidence. And that simpler univariate encoding of confidence then is translated into various behaviors. So it motivates you to give a particular rating of confidence. The studies link the second order confidence judgment with a rating that you ultimately give. But here in Mike's study, we wondered whether this same pathway may ultimately give rise to feelings of curiosity about something. So again, looking at the literature still, we have a good reason to believe, we have good evidence supporting this last step of this process, because many previous studies of curiosity have indeed shown that the curiosity about a topic is related to the confidence regarding that topic. So in these studies, many of them have used trivia questions, including by Matthias, who's listening now, in which people are given a question, and then they are asked to rate how curious they are about that question, and also how confident they are in knowing the answer. And across many different studies, they find similar results, that curiosity is negatively related with confidence. So if you are very confident in knowing something, here on the right of this x-axis, you are not very curious, of course. If you are less confident, if you have intermediate level of confidence, you tend to be the most curious, so intermediate low. And then if you become very, very unconfident, that means that you really have no clue about what this topic is. Your curiosity actually declines a little bit. And so this is actually very good evidence that curiosity depends on the uncertainty of some sort of prior representation, right? If you have no priors at all about that topic, you have no way of defining uncertainty or confidence, and then you have no way of, and then you're not being curious. So this is very good evidence for our hypothesis, at least behavioral evidence for the last step of this chain. But the bigger questions and the bigger challenges we faced in linking it with that very first process. And why is this? So the first challenge is, as I mentioned before, that in most of the tasks of curiosity, these tasks have used trivia questions. So the material that elicit curiosity is a question. So this is lifted from one of Matthias's studies. So for example, a question might be, who was US president when Uncle Sam got a beard? Really interesting. How confident are you that you know this, and how curious are you that you know this? So, so this certainly piques my curiosity. But the question is, for us, we have to have some measure about the uncertainty about this topic, right? So the trigger for our curiosity is this question, who was US president? And we have to have some measure about how is this question represented in the brain, and how do we measure the uncertainty in that representation, right? And at least as far as Mike and I looked through the literature, we really couldn't find any basis for formulating a hypothesis here. However, as I've shown you before, there was work on visual curiosity. So we decided to transform the trivia task just a little bit. And instead of looking at curiosity about verbal questions, to look at a curiosity about ambiguous stimuli. So what we did is we presented to our participants images like this. These images are specialized. They're called text forms, and they're useful because they have been tested in the areas that we are interested in, as I'll show you in the next slides. So here are two text forms. They are distorted versions of some real object. And I wonder if you can, guess what the object is. So let's see, what do you think? Let's look at the object on the left. Any guesses about what that might be? I can't hear you, but I hope I stimulated you to generate some guesses. So it's actually a kitten. So now you can, feel how surprised you are to see that. And now let's look at the object on the right. Any guesses about what that might be? It is a watch, right? So you can see, I think with these examples, I think that they illustrate two things, is that perceptual curiosity is a very natural behavior. We really don't like to see things that are blurry, that we don't recognize. We really attempt to resolve perceptual curiosity. And particularly, and perceptual curiosity and ambiguity or visual uncertainty, we believe is a very strong trigger for visual attention. We immediately orient and try to resolve that uncertainty. So that also motivated my particular interest in it. Anyway, so we use this stimuli in a task of perceptual curiosity. So people saw a distorted image on each trial. They had a little bit of time to look at it and imagine what it might be. Then they rated their confidence about what the image might be. Then they rated the curiosity in seeing the clear image. And then eventually they were shown the clear image. So their curiosity was resolved. And we replicated the results from trivia questions in that there's a negative relationship between perceptual curiosity and perceptual confidence. So if you are very confident in recognizing that distorted image, your curiosity is low. If you are less confident, your curiosity is higher. And you can see that we also have this shift, the curiosity peaks at slightly, not at the lowest levels of confidence, but at slightly higher levels. A little bit of a U-shaped curve there. Okay, so that's wonderful. So now we have a perceptual curiosity task, and we have the behavior handle on that second step. But here again, there's another version of that challenge in that we need to define the representational uncertainty of those images. And the challenge is that all the methods that I talked about from visual research that are tailored for decoding neural uncertainty are really specialized for these elementary features of oriented lines. And the readout algorithms assume that neurons have cosine tuning for orientation, and that's really critical to estimating how much uncertainty is across the population. So this is a challenge number two, which is sort of a close cousin to challenge number one, is how do we measure representational uncertainty for a complex image? Okay, so we know that complex images that correspond to our percepts are encoded in higher order visual areas. And in particular, the occipitotemporal cortex has been intensively studied. And that's a portion of the temporal lobe that is very highly developed in monkeys as well as in humans. And it represents behaviorally relevant object categories. So they're very famous face representations in the occipital temporal cortex, but we also know that there are categorical representations here. So if you look with fMRI during imaging, you can read out whether an object that you see is large or small, and this is in real life, regardless of the visual size of the image. And maybe one of the strongest and most well-documented categories is between animals and man-made objects, or animals and inanimate objects. And those are, when we set up our study of perceptual curiosity, we use the text forms, those ambiguous stimuli, to fall into two categories, animate and inanimate categories. And then, and we found that these categories, as I'll show in a minute, did elicit those categorical representations in occipitotemporal cortex. Okay, so we also carried out a localizer task. So after people did our perceptual curiosity task, we just showed them a bunch of those images that we use, the clear images. Half were of animals and half were of man-made objects. And we find that indeed we evoked the categorical response that we expected in the OTC. And this response was multivariate in the sense that, so here what I'm showing is every little of these colored squares is a voxel, a tiny portion of this big OTC area that we focused on. And the color represents the level of activity, the response in that voxel. And what a multivariate representation is, it is that the average response across the entire OTC was really equivalent for animals or man-made objects, but the particular pattern of voxels that was activated differed. So this is what's called a multivariate representation. This is indeed what we expected. And there are ways, and these kind of representations can be read out with decoding techniques to know whether the OTC cortex thinks that the image it receives is an animal or man-made image. Okay, wonderful. So we have this multivariate representation. Now we have to design a metric of how uncertain OTC is about the category. Again, a metric that does not assume that there is cosine tuning for any elementary feature. And the way we designed this metric was the following. So we took our animal template and man-made templates. Remember, those are the average responses that we expect when we show an animal or a man-made object. And we know those templates from the localizer that we performed at the end of the task. And then we took the response that was evoked by every image in the perceptual curiosity task. And then we correlated the two correlation coefficients. So how correlated was this text form evoked pattern with the animal template and with a man-made template? And then we designed a metric of uncertainty, which is inspired by computational studies of uncertainty, in particular in the machine learning literature. And those studies say that when we quantify uncertainty, we have to think about two things. The first one is, does this neural representation distinguish the two categories? So are the two categories distinct from each other? And here in our, this question is answered by this quantity, which is the difference between the correlation coefficients, the absolute value of the difference, right? So what we care about, the discriminability, high discriminability, is if a text form pattern correlates better with an animal template than with a man-made template, or the other way around, correlates better with the man-made than the animal. It doesn't really matter which way the difference goes, as long as the difference is substantial. It means that this neural representation can distinguish between the two categories. But a second kind of uncertainty is really important. And that is what is called the model uncertainty. And that is does this image conform to either of the hypotheses that you are considering? So after all, here we have a very narrow model. We say something can be either animal or man-made. But of course, there are many more other things in the world, and it's possible that you get an image that doesn't conform to either of these templates, right? So, so indeed, we found that our measures did not work so well if both correlation coefficients were low, which basically meant that this model, our model, doesn't work well for the image that we're showing. And we captured this by the average of these two correlation coefficients, right? So that's the average, the model that quantifies model uncertainty. So our final metric of what we call OTC certainty is the product of these two factors, the average evidence and the differential, the relative evidence. And so that means that OTC is more certain about what it sees, so to speak, if the two patterns are differentiable by categories. It has a good idea what the category is, and if these categories are recognized. So if at least one category seems to fit very well. Okay, so the test of this linear of this measure is whether it predicts anything meaningful in our task, and this was the most exciting result, that indeed this neural measure of OTC certainty is very reliably correlated with curiosity ratings. So if OTC certainty is low, so if OTC is very uncertain about what it sees, the curiosity is higher. And if OTC has higher certainty, people report less curiosity. Okay, so this is great. We now have a link between, we finally managed to bring this first order certainty, sensory certainty into the picture. And we know that there is some pathway that links it to curiosity ratings. And now the question is, right, so now we want to look at the higher order areas that mediate these pathways. And as I mentioned to you before, there are many of these areas, and different areas encode confidence depending on the context and task. So we couldn't really go through a very broad fishing expedition in the brain. But we had good reason to focus a priori on two of these areas that encode confidence. And these areas are, one is the ventromedial prefrontal cortex. And that's an area located sort of over the orbit on the ventral and medial aspect of prefrontal cortex. You might have heard of it. It has been very famously studied in relation to value. It encodes value very strongly. And it also encodes confidence. And in particular, we were interested in it because it has been shown to encode perceptual confidence, so confidence about visual stimuli. And this is in tasks that use very simple-minded, oriented lines, all the way up to tasks that use complex images like works of art and so on. And the second area we were interested in is the anterior cingulate cortex. That is an area that's on the medial aspect of the brain. And that is very strongly associated with cognitive control. So that's including the cognitive control of attention. And there has been one study of perceptual curiosity that didn't measure sensory representations, but showed that ACC becomes activated in relation to curiosity ratings for perceptual images, for visual images. And so we took those a priori regions of interest in the BMPFC and ACC, and we measured their activity in the task. And we found that, so for VMPFC, we found that the average activity in this area scales positively with confidence ratings. So this is consistent with what many other studies have shown. And new to our study, this area scales negatively with curiosity ratings. Which was very good. And this, we found a very similar pattern in the anterior cingulate, right? Okay, so now we have both of these areas are good candidates for mediating the relationship between OTC certainty and curiosity ratings. And to see whether these areas indeed mediated this relationship, we did mediation analysis. And that mediation analysis showed us that the VMPFC in particular, but not the ACC, performed this mediating role. Okay, so here's the mediation analysis. So to give an overview, the mediation analysis tests for the pairwise correlations between different constructs, and then sees if those correlations persist when accounting for other constructs, right? So here we found that the pairwise correlations for all of our three nodes here are highly significant. So OTC certainty is highly positively correlated with the response in VMPFC. So VMPFC could, in principle, read out OTC certainty and translate it into confidence. VMPFC activity is negatively correlated with curiosity, as I've shown you before. And OTC certainty is also negatively correlated with curiosity, as I've shown you a couple of slides back. And now the question is, the key question is whether this last relationship, this C coefficient here, coefficient between OTC certainty and curiosity, does it remain significant after we account for VMPFC activity? And the answer is no. When we account for VMPFC activity, we soak out all this relationship, it becomes null. And indeed, which means that VMPFC activity significantly mediates the relation between OTC certainty and curiosity. In other words, OTC certainty does not directly give rise to a feeling of curiosity, but it only does so by modulating VMPFC activity, which in turn gives rise to feelings of curiosity. So VMPFC is a significant mediator. In contrast, ACC is not a significant mediator, even though it responds very similarly to VMPFC. So again, if we look at the pairwise coefficients, we see very similar patterns. OTC certainty, as I've shown you, is correlated with curiosity. ACC activity is correlated with curiosity. And OTC certainty is positively correlated with ACC activity, meaning that ACC activity could be a mediator, just like VMPFC is a mediator. But in fact, we found when we do the analysis that we don't have significant mediation. So the link between OTC certainty and curiosity is just as robust even when we account for ACC activity. So ACC activity is not a significant mediator. All right, so I think that this brings me to the end of my story. So what have we learned? So I think we learned two interesting things. So first of all, we have a method for quantifying a higher order, the uncertainty of a higher level representation, and we show that that uncertainty is related to feelings of curiosity. And this is exciting because, it might be interesting to think about if we can really think about this higher order. We're starting to learn more and more about how abstract knowledge is encoded in the brain. So can we actually use this metric or variance of it to measure the uncertainty of a higher order abstract representation? And we've also learned that the link between these representational uncertainty and our eventual feelings of curiosity is complex and specific. And for perceptual curiosity, we found that the VMPFC has a very specific role that is not shared by other areas. And it mediates the feeling of curiosity. So another interesting question, I think, to follow up on is what are the specific pathways that link different kinds of material, different kinds of knowledge to ratings of curiosity and confidence. Anyway, so this is all I had to say. I hope I convinced you about why perceptual curiosity is interesting. And thank you very much for your attention.